all over the Hill, we have people who are involved in trying to help members of Congress decide what are good ideas and what are bad ideas and what are the ideas all about at all. So we started some years ago with this Congressional Biomedical Research Caucus because we wanted to try and give people an opportunity to hear what's going on in the world. And we started 20 years ago, actually, with this caucus. And given the wealth of research that's done in this country, it's impossible for us to cover everything, but there are no shortage of leading lights to come here and talk. And they all grab the opportunity to come and talk to you because they know that some way or other you're going to affect, as staff people by and large, you are going to affect what members think about this whole area of biomedical research. One of the reasons for doing this caucus was because we have the National Institutes of Health, and some people don't remember in 1935 or whenever it was when Warren Magnuson put it into law, why it was done or what it's done or how it works. And it's a very complicated place. It's gotten more complicated even in the 25 years that I've been here in the Congress. They have done groundbreaking work with all kinds of things. The most recent one, of course, that everybody knows about is AIDS. And I'm going to introduce Dr. Nadeau, and then I'm going to leave here to go up to the convention center where I'm going to present some awards to people. But if you look at the AIDS epidemic from the time I got here, when it was simply a death sentence in Africa and to gay men in this country, and you look at where we are 25 years later, it is all as a result of this biomedical research real trajectory that is unbelievable, that we should be able to do this with an epidemic in one generation, really. We have really turned it all around. It's not done yet, but it's on the right. It's going in the right direction for us to create a AIDS-free generation in the world within the next few years. Now, I'm proud to be from Washington State and from Washington because it has more than its fair share of AIDS cutting-edge research. It's been involved in a whole variety of places, University of Washington, various institutions in the city, and the Cancer Research Center and the Paul Allen Brain Center. There are all kinds of stuff going on in Seattle. The Gates Foundation is there. So you've got all these people working in this whole area. And finally, there's the Institute for Systems Biology, which was founded by Dr. Lee Hood. The IBS does more revolutionary work in the area of DNA sequencing and personalized medicine and a whole lot of other areas. And having Dr. Nadeau here to talk about that, he's a recent addition. Seattle is a little like a magnet. We draw people from around the country who see all this stuff going on out there in the northwest corner of the United States, and they say, not a bad place to live if you can stand a little gray now and then, a little bit of rain. And it's a very exciting intellectual place to be. He's a pioneer in systems genetics, and he's going to talk today about, and I've got to read it, epigenetic influences passed from parent to child. I wish I could stay and find out what that is. I'm a psychiatrist, and I know that parents have a lot to do with what happens to kids, but I think you're going to hear another side of child rearing, perhaps, from Dr. Nadeau. Dr. Nadeau.
Thank you. So it's a pleasure to be here. My son worked on the Hill with the Olympia Snow for a number of years, and so I have some sense of the kinds of things that you do. So I have a favor to ask before we begin. What I'm going to talk about is quite complicated. For geneticists, this topic drives them crazy because it challenges the fundamental principles of genetic understanding of inheritance. So if you have questions along the way, don't hesitate to ask. Usually you give a talk and then questions after, but if you have some questions along the way, it might help the dialogue. So what I'm going to talk about, the original title was The Sins of Our Fathers and Mothers. But two things with that. First, it's our ancestors, not just our fathers and mothers. Whatever we inherit from our fathers and mothers, they inherited from previous generations. And second, we know about inherited diseases, the bad things that we don't want to inherit, but we also inherit a lot of good things too, and it's the balance of those, the sins and the blessings that we inherit. So what I'm going to talk about today is something that we discovered completely by accident. Someone asked once, why did you look for this? What made you look for this? There's no way anyone, any self-respecting geneticist would ever go out and look for something like this. What you do is you research, and you pay attention to what nature tells you. You pay attention to the results, and you try to be very critical, but also open to the surprises that nature has. So we thought we understood genetics, we thought we understood inheritance, and it turns out we do, but we don't. Remember when you go to court, you're always asked to tell the truth and the whole truth? So everything we know about Mendel and DNA is the truth, but it turns out it's not the whole truth. There's a lot more going on. And it both makes research and our understanding of human biology more complicated, but also more simple. Simple in the sense that the more we understand about what's really going on, the better chance we have of dealing with inherited diseases. So we all understood, even from very early human beings and the curiosity that humans had, that there's these similarities within families, that children tend to look like parents and parents tend to look like their grandparents, and they look more similar to each other, had more features that were in common than randomly selected people in the population. So there was this sense that something was inherited. We didn't know what it was, we didn't know the rules, but this common in the cave, on the street sense of heritability, what geneticists call heritability. Now we've made it more complicated, we formalized the mathematics of that, but if you go back to it, inheritance is simply trying to understand the rules and the molecules that determine who we are from one generation to the next, at least the inherited component. The environment is also important as well. And the first breakthrough was Mendel. He was a monk in what is now the Czech Republic, and he worked with plants, and he would cross things like red flowers with white flowers and get the hybrids that balanced those two genetic variants, the white and the red. And if you intercross these have from each of their parents the red form and the white form. And so in this hybrid, you get the gametes from the male plant and the female plant, one carrying the red, one carrying the white. And when you put them back together in the next generation, you get a red, two whites, and one white, two pinks and one white. So those are Mendel's laws of inheritance. And he showed this for a number of features in a number of plants, in a number of different species of plants. And they explain the rules of inheritance really well. And we've since extrapolated these principles to almost every trait and under almost any species under almost any condition. These are assumptions that we make. One of the problems in research is that the good and the bad, we have to make assumptions to make our work go forward. One of the bad things is that we forget the assumptions we make and we assume that those conditions are true everywhere. It's a truth rather than an assumption. So this rule, what his law enables, is what we call testing the association between genotype and phenotype. Genotype is the genetic information in each of the gametes, and phenotype is the manifestation, the physical manifestation of that information. So that information we now know is DNA, and the phenotype is height and color and disease susceptibility. So it's genotype-phenotype correlation. Now based on these principles, this kind of association between the DNA that you inherited, the genetic variants that you inherited, and the disease risk you inherited is the foundation of all medical genetics. If you have a cancer, we assume, and it's an inherited form of cancer, we assume that if we look hard enough and far enough and deep enough in your DNA, we will find the genetic reason why you have the cancer. And if we can find that genetic reason in your DNA, we can predict that sometime later in your life, you will have the cancer. So it's all about you. What you inherit from your parents came from the past, but the genotype-phenotype interaction is within you, and that's what we test. And as a result of the Genome Project, we're now spending billions of dollars worldwide testing diseases like diabetes and obesity and schizophrenia, all these conditions that we know have a genetic component. We're now looking in affected versus unaffected individuals to find the genetic basis. And with that genetics, we can get at what are the mechanisms? Can we predict with the genetics, and with the mechanisms, can we find ways to treat? So it's both predictive and preventative. But this simple rule is the foundation of almost all medical genetics and billions of dollars invested in research annually worldwide. The next steps were people like Morgan, because Mendel dealt with this as a statistical property. He didn't say what the molecule was. There had to be a physical basis, but we didn't know what it was. So Morgan in the early 1900s showed that there are these molecules in cells called chromosomes, and chromosomes somehow were carrying that information. So now there was a molecule. We didn't know what was in the chromosome that did this, because there were proteins and DNA and other kinds of molecules in chromosomes. But at least there's a molecule. There's something we can see. And then Watson, Crick, and Rosalind Franklin showed that, and others showed that it's DNA, this simple, very simple molecule, simple four-letter code of information, contains all the inherited information that provides us our biology. And then, through the Human Genome Project, Craig Venter, Eric Lander, and many, many, many others established the full sequence of the human genome and the genomes of lots and lots and lots of species. And we had told ourselves, and this is partly true and partly, again, the truth, not the whole truth, if we know the human DNA sequence, then we will know the information code, we will know the information content of humans, and we should be able then to understand human biology. We will know all the parts, all the building materials. The problem is, if you look at, it's like looking at software code. You can look at the DNA sequence and you know a lot, but it doesn't tell us how things work. Genes are turned on or off at particular times and places. The sequence doesn't tell us that. It tells us what that gene is. It tells us what that gene can do, but it doesn't tell us how everything is organized, coordinated, regulated, and controlled. And obviously, control is important. You don't want all of your genes on all of the time or off all of the time. So the Human Genome Sequence and project was unbelievably important, but it's not the end. It's, in a sense, it's the beginning of a long process to understand biology. The other is that this is one of the assumptions that we've made. Mendel never claimed that his laws were universal. People came later and said, well, these laws are true everywhere we look, and so they must be true everywhere, all time, for all conditions. But in fact, on the sidelines of biology, there were all these exceptions accumulating that didn't quite behave the rules. And what we like to do is to set them aside. Well, they're exceptions. We don't really need to pay attention to them. But often in research, it's the exceptions that tell you it's more interesting, it's more complicated, something special is going on. And it might be just that one case that's different, or that might be a clue to something very general. And this is what happened. We stumbled on something on the sidelines that turns out to be probably as important as DNA for inheritance. And this is a field called epigenetics. Pandora let lots of things out in the world. One of them is, in the genetic world, is epigenetics. So we'll talk about that. What is this? It's trying to understand when and where and how a gene is expressed. Because getting that right is really important. How much protein you make, when and where, in what form, determines your biology. If you have too much, too little, this form versus another form, you might be okay, but you might be susceptible to disease. Epigenetics deals with that control. And those patterns of control have to be not only true for that cell, but when that cell divides and makes a daughter cell, you have to inherit that. So it's, in a way, it's thinking about, if I've made a muscle cell and I want to make more muscle cells, I can either let my daughter cells decide how do I make myself into a muscle cell, or I can transmit that control information that says this combination of genes on and off is the identity for a muscle cell, and I'm going to pass that information from mother muscle cell to daughter muscle cell. Who knew? That was hard to say. So you not only have to acquire that epigenetic information, but you want to transmit it to your kids across cell division so that your muscle continues to be muscle. But we recently discovered that this can also be stable across generations. So if there's, in some cases, if there's an epigenetic code that determines the expression patterns for genes in the parent generation, there are circumstances where that information can be passed to the kids. So in a way, the kids don't have to figure out what expression pattern works for them in that world. They've inherited information from the parent. And we'll get back to this at the end. So here's DNA. It's a double strand. It's 4 bases, A, T, G, and C. They pair up in a specific way. part of the DNA is genes. Maybe only 3% of all of the genome is genes. The rest of this was thought to be junk, but usually in biology, when you say it's junk, probably means it's got some other function. We just don't know what it is. You'd have junk at home. Sometimes some of it's useful, right? Just don't know what it is yet. And then these are embedded with lots of proteins into chromosomes, and chromosomes sit in cells, and cells compose who we are. And when a gene is transcribed, the DNA unwinds a little bit, exposes the gene that you want to make a copy of, and then that makes a messenger, a transcript, a copy of that gene, and you can make many, many, many of those. And then those are translated into proteins, and proteins are the building blocks. Proteins are what you use to build biology. So this is the way to translate information into function. And this is the step that's controlled by epigenetics. And there's two ways. There were two primary ways, but we'll discover that there's probably a third way. The two primary ways are something called methylation. You can add a carbon group to particular sites in a particular base, so it's cytosine methylation. So if you methylate that in particular numbers, in particular places, in particular ways, the gene next to it will be turned off. If you demethylate, the gene can be turned back on. So it's not sufficient to turn on the gene or off the gene, but it controls a fundamental step. If it's methylated, you're probably not going to make that gene. So what controls methylation, how this works, not clear. The underlying DNA sequence is completely the same. So you can look at the DNA and not know whether that site is methylated or not. You need to do an epigenetic assay to ask about methylation. There are probably many other chemical modifications that we simply don't know about, and there's some new technologies coming out that will let us interrogate those. But it wouldn't be surprising if the fundamental code of DNA is modified chemically in ways that determine things like expression patterns. There's also another way. Your DNA in the cell isn't just naked DNA floating around. It's packaged, it's wrapped around proteins called histones. And histones have these long tails, a long little strand that hangs off the end. And those can be chemically modified. And depending on the nature of these modifications, the gene can be turned on or off and determine the level, time, place, extent of gene expression. So DNA methylation, histone modification are two fundamental aspects of controlling gene expression patterns. Okay, what did we discover? We began by talking about Mendel. Yep. The three. We're going to come to the third one. They're RNAs. So if we go back here, these RNAs and some other small RNAs that are a little bit unconventional, they're probably involved too. But we'll come back to that later. That's okay. Those are the kinds of questions that are good. You should ask when you have them. Okay, Mendel taught us about genotype-phenotype associations within individuals. What we've discovered in many different conditions in laboratory mice, and we'll talk about why laboratory mice are critical for these discoveries. The phenotypes, the disease risk is inherited, but the genetic variant that caused that condition, that changed the risk, can be present in ancestral generations. You don't need to inherit the genetic variant. You need the variant to initiate an epigenetic change. That epigenetic change is then transmitted on its own, independent of the original gene. So you inherit the disease risk, you inherit the phenotype, but not the risk. You can look then in the DNA of that individual and never find the genetic reason for the condition. There was a genetic reason, but it's in previous generations. And this profoundly changes the way we think about inheritance and the way in which we do studies to understand disease risk. So there is a genetic basis. It is familial because it's going to follow in the descendants of that individual that initiated the condition. It's going to have all the feeling, all the things that you look for in a conventional way about inheritance, except we can look in the DNA of that individual forever and never find the reason. Okay. I'm going to give you 3 examples of this. One in testicular cancer, one with a Y chromosome in the daughters, and one with obesity. So when I started in research as an undergraduate, I worked in Roy Stevens' lab. He discovered testicular germ cell tumors, the testicular cancer, most common solid cancer in young men. It's readily treated, but there's still a lot of the biology that we don't understand. And they say it's readily treated, and it's true, 95% recovery rate. But if you're a young man with testicular cancer or someone is in the family, there's chemotherapy, it's surgery, there's metastasis. It's really, really hard. So it's cured, but it's not really cured. We ought to be able to find better ways to treat a condition than surgery and chemotherapy, which almost kills people. We should be able to do better in medicine. In the 129 family of inbred strains, these tumors occur at a measurable rate. In other strains, they're rare. In this strain, about 5 to 10% of males, they're all genetically identical, they're all inbred, they're all cloned, as it were, show these tumors spontaneously. So there's no carcinogens, there's no mutagens, there's no special manipulations. It's just, if you look at enough animals, you find an affected mouse. And the tumors are interesting. They come from the germ cells that will eventually make gametes, that will eventually make sperm for the next generation. What does the sperm have? What does the sperm and egg have? It has the potential to make a whole organism, right? It has the information and is primed to make the muscle, skin, hair, bone, all the biology of an organism. In these tumors, it's as though the stem cell gets a signal saying, oh, here's the switch, make an organism. So in the tumors, you see, in a muscle tumor, you see muscle cells. In a brain cancer, you see brain cells. In these tumors, you see muscle, skin, hair, bone, cartilage, retina from the eye. You can find teeth, you can find hair, sebaceous glands, but it's all disorganized. You can find brain next to small intestine. You can find teeth next to stomach. So it's like, I know how to make cells and tissues and body parts, but I can't organize it right. So it's really remarkable. They're fun tumors to look at because you never know what you're going to see. One day I was looking at a tumor and it was doing this. It had made cardiac muscle, the muscle had organized and it was beating. And it had made some blood islands trying to make blood in a little vessel. So in this tumor, in the testis, it's a really, really interesting biology. What do we know? It's genetically controlled, but it's, but it's really complex. There could be as many as 1,000 genes that control susceptibility. When you get to that many, you wonder, you want to know the genes, you want to know their functions, but you wonder, how much am I going to learn if there's 1,000 different genes that are involved? So not only the logistic problems of studying the genes, but then what's the consequence? What will be the benefit? We know the stem cells are germ cells, that I just told you. We know to the day in these mice when the critical period is. There are cells before embryonic day 11 and a half and 12 and a half, and there are germ cells after that day, but something happens during that day that's critical. Now they're going through some developmental changes, so something in that, what those cells are doing during that single day is critical. If those cells get through that day, okay, they'll be fine. If they have problems, then in these mice, months later, and in humans, years later, decades later, you'll see a tumor. So the tumor's here, but the critical event was early in development. And in mice, we can see the tumor at 3 to 4 weeks of age. If they're going to have one, they'll have one by that time. This is the simplest example we have of these transgenerational effects. And we'll contrast, and it works in males, but not in females. So I'll show you the females first in the conventional Mendelian way, and then we'll see the unconventional effects. So this is a gene called KIT ligand. It's involved in signaling between cells. Cells, we're not just a bag of cells. The cells have to coordinate and talk with each other. And KIT ligand is expressed from one cell, secreted from one cell, and passes to other cells to communicate information. This is a mutation, it's a deletion, so these mice don't have this ligand. So instead of having 2 copies, 2 doses, these heterozygous mice, like this, have only one. So it's not a mutation that's doing something weird and strange, it's simply a sensitivity to dose. I need 2 copies, but I don't have 2 copies, and given half what I should have, cancer risk is going to change. So if mother is heterozygous, and the female is always listed on the left, is crossed to a regular male, a wild-type male, we're only going to look at male progeny because these are testicular cancers. By Mendel, we get heterozygous kids and wild-type kids, half and half. Mendel would love this. These show the baseline rate of 5%. These show an increased rate of 15%. So simple partial loss of function increases risk 3-fold. It's not huge, but if you look at a lot of cancer risk, genetic variants often act in that kind of way. A 3-fold effect is pretty big. If it was you, it would change the way you think about your risk. So this is very conventional inheritance. If you know the genotype, you know the risk. If you know the risk for the group, you know the genotype. The genotype-phenotype works. If we reverse the cross, and now dad is heterozygous for this, we get exactly the same 2 kinds of kids. And in these animals, the risk is increased. And for years, people have studied this, trying to understand, why does partial deficiency increase risk in this way? What people ignored was the wild-type control brothers of these individuals. In the brothers, they're completely protected. We have never seen an affected animal. Now, by genotype, these two individuals are exactly identical. They're cloned. There's no genetic differences between them. But there's a big difference in 5% risk versus, which is 1 in 20, versus complete protection, 0 risk. And here, if you want to know the genetic information that determines the risk, it's not in their DNA, it was back in the father. So it's the previous generation that determined the risk, not the genetics of that individual. And we don't know yet, we're studying, what is it that's inherited, that's not DNA, that's providing this protection? That would be pretty interesting. If we knew epigenetically how to protect, maybe we could therapeutically protect. Okay, given those results, we had an idea about another gene called ApoBec1. We're not going to talk about the biology. The biology is less important than the principles. But we did the same thing. We made a knockout of this gene. It's deficient. So this mouse is partially deficient. And by the time we got to this case, we knew that keeping track of paternal lineages or maternal lineages was important. So the same kind of cross, heterozygous dad to wild-type mom, generation after generation after generation. We get the same two kinds of kids, heterozygous and wild-type. Mendel would love this. 26% of these are affected, 5% of those. So a 5-fold increased risk simply by a partial deletion. And it's what I'm telling you, these genetic differences are the only differences between these animals. They're genetically identical, except for, in this case, this gene. The other ones in the previous, they're genetically identical except for that partial deficiency of KIT ligand. And part of the trick in figuring this out, we'll get to later, but it involves creating deep genetic uniformity. Okay, so this acts in a conventional way in the paternal lineage. Look in the maternal lineage. So mother to daughter to granddaughter, all of the kids are completely protected, regardless of their genotype, regardless of the genetic risk they have. So it's the same genetic variant, it's the same dosage here in these two. When inherited from the father, and we're looking at the sons. When inherited through the father's lineage, the risk increases. When inherited through the maternal lineage, all of the kids are completely protected. And again, the only way to know the risk is to know the genotype, the genetics of ancestral generations. Now, the students who worked on this took these animals and crossed them together, the wild-type animals. Now, these are normal 129 mice, and we crossed them together to ask, does this effect persist for one more generation? Or is it directly parents to kids? Guess what? The kids are protected. So they went another generation and another generation. And as long as we bred them, those kids were protected. So in this particular study, it was great-grandmother's genetics that determined the protection of the great-grandson, the cancer risk in the great-grandson. Nothing in their DNA would tell you. It was something generations before. It was ancestral genetics. So if there's an environmental component, as we're going to talk about, or a genetic component, it means we need to understand either directly what happened in ancestral generations, or more likely, we need to be able to interrogate, catalog epigenetic changes that the individual inherited to understand disease risk. We want to know the genetic variants, but we also want to know these epigenetic features too. So this is pretty cool. Whoops. No one's ever seen anything like this before in cancer risk. And we sort of know from cancer biology and Genome Project and all the studies that are ongoing that we're finding the disease genes. But if we add up the individual effects, the individual risk are that's accounted for by individual variants, is pretty small. If we add up all of those risks that we know about, if we get to 10% of the risk, we're doing really well, which means that 90% of the risk is still unexplained. And what we suspect is that some things like this, it's inherited, it's genetic, but it's not interrogated in the way that in conventional studies, and so we miss it. You had a question?
So are they independent of one another, or can like the maternal suppress? Is one dominant? Does the maternal suppress the paternal line, or vice versa?
Oh, you can. Yeah, it is suppressed. That's a good question. I hadn't thought of it that way. But you're right. In this case, the maternal line is suppressing the paternal effect, because the paternal effect here is 5%. And remember, one of the wild-type alleles, the normal alleles, is coming from mom, and one's coming from dad, and mom wins. Probably always the case, right? Mothers always win, females always win. That's what we tell ourselves anyway. But that's a good question. I hadn't thought of it that way. And whether that's a general property or it depends on the circumstance, depends on the biology and the genes. I don't know. But in this case, mom wins. Okay. Daughters of fathers with different Y chromosomes. We wanted a circumstance where there are individuals who are genetically identical but don't inherit the genetic variant that initiates these conditions. So that sounds a little weird because usually we get DNA from our parents, but there is an exception. The daughters don't inherit their father's Y chromosome. If daughters inherited their father's Y chromosome, the sex-determining chromosome for males, then they'd be a son, not a daughter, right? So that's a case where there's a genetic variant in a chromosome, a whole piece of DNA in the parental generation that half the population doesn't inherit. And so if there are epigenetic effects of the Y on the daughters, or if the daughter's phenotype depends on the Y chromosome, then it would have to be epigenetic because there is no inherited chromosome. And what we wanted to know here is, are these effects strong or weak? Are they common or rare? Because we found a few examples, but that doesn't tell us the generality. Are these little annotations that still sit on the sideline of core Mendelian inheritance? Or is this something in play that could be as important as Mendel? So years ago, Eric Lander and I, Eric is at the Broad Institute now, he was at MIT and Harvard, took 2 strains of mice and moved chromosomes through crossing, replacing a chromosome in the host strain with a corresponding chromosome in the donor strain. We had reasons to do this, and we won't go into it. But here's the black 6 strain, here's the AJ strain, and one at a time we moved each chromosome from black 6, from AJ, onto black 6, replacing that corresponding chromosome. So the new strain, like this one, looks just like black 6, except it's got chromosome 1 from AJ, or chromosome 2, or 3, or 4, or the Y. So now in this case, with this strain, we have males that have different Y chromosomes, a black 6 Y chromosome, or here an AJ Y chromosome, but the rest of the genome is from black 6, and the daughters are identical to each other genetically. The only difference between those daughters is the father's Y, that they don't inherit. So we thought this could be an interesting place to test this problem. So we looked at lots and lots of traits. Did I lose something here? Oh, it gets back to something I alluded to earlier. These kinds of problems are really, really, really hard and maybe impossible to detect in humans because what's the source? We see variation among us. There's 3 sources of variation at least. One is genetic, one is the environment, what you eat and what you drink and the air you breathe, and the other is epigenetic. If all of those are in play at the same time, in a heterogeneous population in any view. If we want to know what's the relative importance of genetics, environment, and epigenetics in the height differences in this room, we can't do it. There's too many variables. And so they're probably all in play, and we want to know the relative importance. And it's really, really fundamentally important to be able to do that, but we can't in a heterogeneous population. In mice, we can control the genetics. We can make animals genetically identical except for particular variants. We can control the environment, and therefore any variation that's left must be epigenetic. If it's not genetic and it's not environment, it's got to be epigenetic. So we looked at hundreds of traits, here 120 traits, and all kinds of blood chemistry and heart functions and response to high-fat diet, all kinds of things. And what we wanted to know is, do those identical females, daughters, differ? If the father's Y has no or negligible epigenetic effects, those females should be pretty similar. If the father's Y affects the daughter's phenotypes, then they could differ quite a bit. So this was the design we took. We call these strains consomics when they have a different Y chromosome. So here's black 6 with its Y, making black 6 homozygous female daughters. And we call these pseudo black 6s. The only difference in these females, genetically they're identical, the only difference is the identity of the father's Y. And we did another strain combination here. And as soon as the females were pregnant, as soon as the male had impregnated the female, we took the father away. Because males with a different Y chromosome might have different behaviors. Those behaviors might stress mom. Mom's stress might stress the fetus. The fetus, months later, might have different biology, right? And if a pregnant mom, women here who've had babies know that probably the father in the house can cause some stress. As soon as the kids were born, we took the brothers away because the brothers have different Y chromosomes. And women in the room who have brothers know that they can be a source of stress. So what we wanted to know is not the social inheritance, but the epigenetic, the biological inheritance. So here's all the other chromosomes that we substituted, and we looked at the fraction of the 120 traits that differed and the effect sizes. How big were these phenotypic differences, say in blood chemistry or obesity? And you can see that about a quarter to a third of the traits differed in the daughters and the males, and the effect sizes were pretty big. The trick is, the big question is, what about the daughters, the black 6s and the pseudo-black 6s? Here's the percentage of traits, and there's the average effect size. The percentage of traits is indistinguishable, basically, from having inherited, substituted a whole chromosome in a conventional way, and the effect sizes are as big. So across this panel of 100 traits, more than 100 traits, epigenetic effects turn out to be as common and as strong as conventional inheritance. So that means there's a whole lot of biology. It means these aren't sideline effects. These are probably central in biology. And we've been missing this. How this works, what it's doing, what the molecule is, we don't know yet. But what this says is probably quite important. Okay, the last, diet-induced obesity. You all know this statue, David right, in Florence. So this is David before he went on sabbatical to the US for a couple of years. And here's David on his way back. So the same DNA, and if he had stayed in Italy, presumably, this would have been David after sabbatical, but, right? And what we want to know, and this is a huge, huge problem. Diet-induced environmental impact on health is huge. It's estimated that a substantial fraction of the world's population is going to be diabetic in 20 years or so. We can't afford that medically, economically. That fraction of the world and all the health consequences to the individual, to the family, and to the economy for that much of the world being diabetic is crazy. We have to do something. And it's not so far between now and 20 years when a substantial fraction of the world's population is diabetic. We already know that the fraction of obesity and overweight is really high. Something like 2/3 of the US population is already overweight. Not far behind, and children as well as adults. Not far behind that are the metabolic consequences of that, like diabetes and liver disease and liver failure. And this is going on worldwide, so we've got to do something. So we're trying to, in our lab, we're trying to find ways that prevent diet-induced obesity, eating a high-fat diet and having funny lifestyles, compromising lifestyles leads to this, or to study the genetics and physiology of weight loss. And we're making really good progress on those. But that's the holy grail in this kind of research. So here's two strains of mice, black 6 and AJ again. The red lines are AJ on a high-fat diet, and 58% of the kilocalories come from fat. So it's like if we took this room for 100 days, you could, whenever you want, as much as you want, as often as you want, you could have a Big Mac with cheese and a supersized cola. So in general, your physician is right, not a good thing to do. But I guarantee if we did this in this room, most of us would show what the physician predicted, but some of us would be okay. And AJ, you can see, is eating the diet, but not gaining much weight. Black 6, on the low-fat diet, gains a bit of weight. On the high-fat diet, it's 40% more. So over that 100 days, it's obese, it's diabetic, it's heart disease, it's hypertension, liver failure, and if we let them go longer, eventually liver cancer, all the metabolic features. And so we're trying to understand the mechanism of disease. AJ mice are pretty interesting. What would you guess? Are they eating more or less than black 6? Remember, they're lean. So what would you guess? What would the prediction be? They're eating less, right? But in fact, they eat 40% more per day than black 6. So it's not that they're eating less and staying lean. They're eating more and staying lean. So then you say, okay, well, they must be active. They must be running and exercising it off. So you'd say the AJ is the active strain. They sit in the corner and they get up to poop, pee, eat, and drink, and then go back in the corner and sleep. Black 6 is around the cage like this all day long. So this is the athletic mouse, and it's got the metabolic disease, and this mouse is the American solution to the problem, because we know we should eat better and less, and we know we should get off the couch, but it's completely protected from all of the diet-induced conditions. So these mice are important, black 6. But AJ is saying there is a genetic solution to this. You can have your McDonald's and Burger King, and you can have the couch and stay healthy.
I was going to ask, are B6 and AJ different genetically or just epigenetically?
They're different genetically and epigenetically. These are two randomly selected inbred strains. They weren't made to be anything in particular. It's like taking two people at random in the room, cloning you, and getting IRB approvals to do whatever we want. which could never happen, thankfully. And it's just, they happen to differ in the response. Now, Black 6 is what usually happens in humans. AJ, we all know this. We know people who eat a lot and don't exercise and stay healthy. Doesn't seem fair, but they're saying there is a solution to this problem. And again, the exceptions telling us that there's something interesting going on. Okay, so we went back to the chromosome substitution strains and we put each of them on a high-fat diet for 100 days, high-fat versus low-fat. And 4 of the strains stayed obese, like Black 6, and most of the chromosomes, most of the strains, chromosome substitutions, were lean. So they were protected from the metabolic diseases, which means there's 18 different solutions to this problem. So trying to find the genes, we took one of the chromosomes, chromosome 6, and we break it into pieces, and we start asking, okay, so it's here. Where in that chromosome is it? Oh, it might be here. Let's break that apart, and where is it? Where is it? So in this one, which has a really big effect, we call it, the gene is called OBRQ2, we took broke this into 3 pieces. It turns out there's 3 genes here, not 1. But we're going to work with this strain called 161A, and this is randomly selected. We didn't know anything about the genetics. We didn't know anything about the biology. There was no evidence in anything we had done for epigenetic effects across generations. It was just, a postdoc had a hunch. And I said, if it's me, don't do it. This is crazy. You're talking about me spending a year's worth of your time, my time, money for mice and food. And food is $2 a day per cage of mice. It's crazy. But he came back in a year. I don't tell my students not to do things, but we talk and said, I counsel them, maybe not this one. He came back in a year and did this. And I said, okay, you're a senior author. The student you work with is first author. I'm in the middle because you figured out things I did. And it's really cool and really important, so watch. Here's body weight between Black 6 and 161. So each dot is a mouse after 100 days on the high-fat diet, and the bar is the mean. So you can see a big difference in body weight, a big difference in insulin levels, and a big difference in glucose levels. So here's obesity, whoops, and here's pre-diabetes. They're called insulin resistance, and it will have to go long enough, they'll be diabetic. So there's a gene in here, at least one gene, that controls obesity and diabetes. So what he did was to take that strain and cross it to Black 6, so made the hybrid, just like what we did with Mendel at the beginning. Remember, the only difference between these mice is that little randomly selected 3 million base pair segment. He intercrossed these, just like Mendel did, and you reconstitute Black 6, and you reconstitute this strain. We're going to forget the hybrids. And if everything is conventional, if everything is Mendelian, these will be obese, and these will be lean, right? 5th grade genetics, 5th grade biology. All the mice are on a standard, it's important to remember, all these mice that are bred are on a standard diet. What we want to do is test their response to a high-fat diet. So we breed, and when we want to test, we take some of the mice from the breeding colony over here, we put them on the high-fat diet and ask how they respond. But these mice are never bred again. What we want is the inherited susceptibility to response to a high-fat diet. If we had them on the diet, then we're mixing the genetics and the diet and we don't know what's going on, what's primary in controlling this. Okay. Here's Black 6 with Black 6 parents. This is body weight. And here's the congenic strain and the difference between them after 100 days on the high-fat diet. And then these are the reconstituted congenics. And they're lean like their parents, or their grandparents, right? And there's the p-value, the probability that it's chance. Here's reconstituted Black 6. Now, the reconstituted Black 6 and the real Black 6 are genetically identical. We could sequence their DNA, and there are no differences between them in the sequence. But now these are lean compared to the parents. So in the DNA, if you sequence the DNA, you'd say these should be obese in response to, show obesity in response to high-fat diet. In fact, they're lean. So knowing their parents' genotype is more important in determining, predicting the risk than their own genetics. So then, Dave, Buckner, and Soha, the postdoc and the student, intercross these just like we did earlier. These are regular Black 6s, so we intercross them and ask, does this effect go to the next generation? And it does. And before we left Cleveland to go to Seattle, they tested one more generation, and they're still protected. And my guess is, as long as we keep the environment and the genetics constant, these mice will be protected, from high-fat diet effects. Now, in this case, it affected the obesity risk, but insulin glucose is the diabetes was inherited in a conventional way. There's a difference in the biology that we don't understand. So another example, a common example of a disease and transgenerational effects. So when we know about the epidemic of obesity and metabolic diseases in the US, in the world population now, we think of what are we exposed to? Maybe it was our parents or grandparents or great-grandparent exposure that was critical. And we can probably never go back and find out what those factors are. But what we need to do is to figure out how to interrogate epigenetic changes in the genome that get back to this, that address this. So we know from testicular cancer that the effects are strong and they persist for generations, and it's transmitted through females. Through the alternative Y chromosomes, we know the effects are strong and common and through males, and the diet-induced obesity and feeding habits at least 3 generations, if not more. So the implications of this, why is this important? First, it says DNA is important, obviously, because it initiates the condition. It confers lots of conventional inheritance, but there's at least one other molecule that's not DNA that's inherited that's mediating these effects. And when we say inherited, it means it's in the gametes, it's in sperm and eggs. The dogma we've been told is that what's in sperm is DNA. Well, it turns out there's RNA too. And what we think from other genetic studies that we've done, you asked for the third molecule, what we suspect is small RNAs. For years we've known about large RNAs that make proteins, but there are a lot of small RNAs that turn out to be regulatory. And what we imagine is that conditions in the previous generation set up a particular set of regulatory RNAs that are packaged in the sperm or in the egg and transmitted to the next generation. So we want to know what's this molecule, because that would be pretty interesting. What's the molecule that's not DNA that's inherited and determines your disease risk and your phenotype and your biology in ways that we know that DNA can? And how do we test for inherited risk? A friend of mine who's very prominent in NIH now, when I told him about this result a few years ago, he said, if this happens in humans, we're screwed. And I walked away thinking, oh, because it's part of the problem is that we create a world around us and we make assumptions and we include and exclude things arbitrarily. So he's right, we're screwed if all we ever interrogate for disease risk are DNA sequence differences. Obviously, organisms, mice, and there's evidence for this in plants and other species, the organisms are inheriting a molecule that's determining the biology. It's not magic. There is a molecule that's doing this. If we knew how, and they're using that molecule to change their biology. It affects their disease risk and how they function. If we knew how to interrogate that molecule the same way the organisms do, then we could do DNA sequence changes and these epigenetic molecules and predict disease risk in the same way. We just need to expand the way we think about biology and expand the world that we interrogate. And then treating and preventing. We've all talked about gene targeting, gene therapy. Maybe there's epigenetic targeting. Remember, this isn't changing DNA. This is changing the way we express genes, when and where and how much. That might be easier to do. It might be harder, but at least it's created a whole new way of thinking about treating inherited conditions. Okay, what do we think? Now, why did I switch this to sins and blessings? We typically study disease as dysfunction of genes and as problems. But always, the other side of it is that gene has a normal function. Normally, it's doing what it should to give us good biology. Most of us are healthy most of the time. But the abnormal part is what we focus on, we pay attention to. So in this case, and people have written this is just an aberration of epigenetics and there is no normal function for any of this. I don't believe that. What I think is happening is, and I alluded to this a little bit earlier, is that the genetic and physiologic conditions in the parents determine an epigenetic code that works for them. In this environment, with my genetics, if I control expression patterns in this particular way, I'll be okay. Now, if I could transmit that epigenetic code, that information, that regulatory sequence to my kids, they would be pre-adapted to that world. If they happen to be in the same environment and the genetics is probably similar, then they might have a head start adapting to the world compared to somebody who's got to figure out on their own. What genes do I need to express and when and how much and where and right? So you can imagine that the real function is to be able to let offspring have access to a regulatory regulatory information that's appropriate for them in that environment. And it's dynamic, it changes, it can change. So think of it this way too. If we run up and down stairs, if we do exercise, if we have a big meal, we have to do something called homeostasis. Our biology changes. If you exercise, you have to breathe harder, move more blood, deliver more oxygen. When you're done running and you cool down, you go back to normal. So homeostasis is there's a perturbation and you return. Your DNA doesn't change, your biology doesn't change, it's just you adapt to the world around you by homeostasis. Over really long evolutionary periods, what do? If the environment has changed, we change our DNA sequence and fix it so that we've adapted to the world and all of our kids will. The chance of going back is almost nil. So we get within a lifetime and over long periods. What do we do over short periods? Now this is partly where the geneticists and evolutionary biologists go nuts, because we don't talk about that next couple generations. We sort of assume that it's boom, you're doing yours and then it's evolution. But what if there was this way of transmitting information from one generation to the next in a dynamic way that can change? Remember, epigenetics is changeable. DNA sequence isn't. Physiology is dynamic. If the environment persists, then that epigenetic code might be good. If the environment changes, the organism could say, okay, I'm not going to change my DNA sequence, I don't need to, but I need to adjust things in this way to adapt. Or if the genetics in the population changes, I need to change things in this way. So it provides flexibility and adaptability and balances some inherited information. If those conditions persist, then I change the DNA sequence and make it permanent. So it's this way to transition from complete homeostasis to completely fixed in DNA sequence. Okay, these are the people. This work doesn't happen without lots of really smart graduate students, really smart postdocs from all over the world. It's always amazed me that kids from other countries, other cultures, other places are willing to leave home and family and language and culture and come here to study and learn and work. And these kids are from everywhere, and they're off sometimes back home and sometimes here doing incredible research. So I'll finish, and if you have questions, I'd be happy to answer. If any of you know, do any of you know Greek? Anyone know that? That's Greek for thank you. No questions?
Yeah. What role might epigenetics play in the rising incidence of autism?
Genetics, a lot. Epigenetics, almost certainly. I've been trying to get access to some families and information to test this in autism cases. It would be shocking if there's not. But getting access to enough materials, the right kind of materials to test that is really hard. Conventional genetics is hard in autism. Unconventional genetics is harder still. But I wouldn't be surprised if there's, what we're going to try to do is look at some mouse models to get a feel.
Are there any companies yet looking at epigenetic therapy?
Oh, sure. Absolutely. Do I know what they're doing or where they are? No. But yeah, there absolutely are. Around cancer, around behavioral, neurologic conditions, probably around metabolic conditions. Will they be successful? Probably not. In the sense, because most startup companies, 90% of startup companies fail, and we hear about the successes. And it's still very, very, very early days. This is really complicated biologically, because what you want to do is you don't want to change the whole epigenetic code. You don't want to change methylation across the whole genome, because many of those methylated changes are right and adaptive, and those are just what you want. What you want is to change methylation here in some specific way. We don't really know how to do that yet. We know how to do the whole genome, but then the mice are really sick. We know how to make histone changes, but the mice are really sick across the whole genome. What we need to do is figure out how to make this specificity. So genes have a specific sequence. We can target that. Histone changes, methylation changes, RNA changes. We haven't figured out the specificity yet. So I wish them luck, but my guess is it's going to be really hard.
How much more complicated has this now made the whole study of genetics? I mean, it was sort of complicated already, but now my brain hurts.
So a friend of mine who's a geneticist, I'm a geneticist, and a friend of mine, geneticist, once said, why did you look for this? Well, you don't look for things like this. And second, he said, you're trying to destroy the field of genetics. I said, well, first I have a vested interest in not doing that. And second, if that's the biology, if that's the way it works, then we have to deal with it. So it does make it more complicated, as you said, it makes your head hurt. But the other, when you look at it, you say, oh, that makes sense that it would work that way. Because if you tried to build an organism and let it be adaptable and flexible and all these other things, without something like epigenetics, how do you do that? If you're an engineer, how do you make an organism that way without epigenetics? So it's got to be there. We can't explain biology without it. So it's both complicated because it introduces all these other questions that we didn't think about at all. Or if we did, we sort of went, you know, yeah, we know, but we don't want to know. To now we're going to have to deal with it. So it both simplifies and complicates. Yep.
I'll follow up on that, because it seems like in the development of biologics and treatments that are based on genetics, that there's frequently a fairly low success rate among the population. I mean, some people are benefited and then a lot of people aren't. And could the epigenetics of it determine who is benefiting from the biologic treatment?
So in another talk I give, in a longer talk, there's two things that come out of Pandora's box. One is epigenetics and the other is epistasis, gene interactions. All the models, until very, very recently, deal with genes as individual entities. If we know this gene, this is what it does, and it does this, it has this function in all times and all places. And it turns out that's probably completely wrong. Eric Lander and I had a paper a couple of years ago looking at this issue. And it's probably completely wrong. I don't think we found any genetic variant that had that simple, constant, predictable effect. Everything was context dependent. It has this effect depending on the rest of the genetics of the organism. If the genetics is one way, it'll do this. Like the OBRQ2, that's almost as strong, has almost as strong effect as the whole chromosome, and almost as strong as all of the other AJ-derived genes on resisting diet-induced obesity. That one little segment. The problem is, if I make that segment one little bit longer, there's something here that completely hides that. So I could look at that segment and say, oh, protected, cool. I put this in, that effect is completely, completely gone. So if you put epigenetics and gene interactions, epistasis, together, then it starts to deal with this complexity. It's not as simple. We hoped it was simple. We hoped that complex diseases are simply Mendel writ large, a whole lot of Mendel over and over and over. Again, you don't build organisms that way because the genes have to work together. That means they interact. If they're doing Mendel, it means they're not interacting. You could look at each one and tell what they do. It doesn't work as well. You had a question?
Yes, I was just wondering, the research you've done, I'm wondering if it was already published or it's to be published?
A lot of it's already published. There's, do you know about PubMed? Okay, do a PubMed. Okay, do a PubMed with my name, and then you'll find some reviews that we've done, and there's some other papers in press, and we're still doing more work, but some of it's been published. Yep. NIH had this a little anecdote how NIH works. When we first found the evidence for testicular cancer, I called the program person at the National Cancer Institute and told her about it. She said, oh, everyone's already saw the paper, and it's the coolest thing we've read. It changes everything. It's the most important paper, and so this is nice back and forth. So I'm going to put this in my grant renewal. And she said, oh, that's too bad. I said, well, why is it too bad if it's the coolest thing you've read in years? And she said, because it's completely unfundable. I said, what do you mean it's unfundable? And I knew the answer, because study sections won't like it. There's no place, as you said, it makes your head hurt. There's no place in their head to park this. So with funding levels the way they are, they fund safe things. And these things that sit on the side, no. So she says it's completely unfundable. So NIH a few years ago, maybe 8 years ago, started a program called Pioneer Program. One of the criteria is the work has to be original and novel and innovative and unconventional and all that, and unfundable through conventional routes. So I quoted her in my presentation, and it worked. Yep.
I have a question just following up on just that comment that you just made about the funding structure. And this is the same exact question that I asked the previous speaker in the series that came a couple weeks ago. And that is, do you think there needs to be a change in the, not the practice of science, but the culture of science? For example, with grant funding, or this whole rat race of how many publications you have before you go up for tenure, or that you have to have a paper in Cell Science or Nature. Or I used to be in this field. So
Yeah. I can tell.
It's like a checkoff list. You gotta have.
Sure.
And everyone, they all claim, well, we don't look at that, this and that, but that's, I
Yeah,
don't believe that.
yeah,
I mean, there's
yeah,
a lot of social
yeah,
pressure to look
yeah,
at
yeah.
things that are.
Yeah, and especially,
So anyway, I just wanted to ask you if you had any.
especially as money gets tight, what do? You become more conservative, more conventional, safer. It's natural human instinct to do. If money is tight, are you going to fund more unconventional things and leave the core of what you think are the essential aspects of biomedical research unfunded? You're going to put those at risk? It's human nature to protect the center. And so having these other programs around to deal with unconventional is good. You're right. I sit on review groups and advisory groups. And we tell, there's special programs for young investigators, and we tell them that the funding, that the review criteria are more relaxed because we know that they're new and they don't have the preliminary data and they don't have the track record. And everyone in the review group is reminded of this before they review those applications. And then you get the application. Well, they don't have the number of publications we're looking for. We don't have the, it's human nature. And these are, and academics will say, well, the review groups, we hate them. They're incompetent and they're biased, but they're us. It kills me when people say that. I don't know. How do you fix human nature? It's funny, you can get a room of people, you can get individuals outside of the room and they're completely sensible, thoughtful, practical, and you put them in the room to review grants. But it's not just in medical research, it's what we do.
In Congress, too.
And it's what we do. We're really good at doing that. I don't know how to fix that. Well, one way to fix it is to have more money in the budget, but that's a fix for everybody, right? Yeah.
I have never studied science, and biology was not my forte at school, so this is a complete layman's terms. My uncle has motor neurons. My grandfather had motor neurons, and it's genetic. I'm not sure if it's an inherited thing that's coming, or it's just been a freak accident that both.
Or epigenetic.
I have no idea exactly. So. What do you think the dangers of epigenetics are? Because for me, I see it as 20, 30, 40, however many years in the future, people will be able to get their children tested, or people tested and say, right, well, it says that you have a high probability of getting cancer of X, Y, and Z.
Yep.
Can that not present a danger to human and mankind as an entity that we are able to say, well, might get cancer when I'm 40 or 50, so let's remove that, tinker around with it, or if we know in the future what.
So this is a
what do you think the dangers are for this?
It's both. So epigenetics is both a danger and a risk. There's a good side and a bad side. If the normal function is to pre-adapt offspring to the particular genetics and environmental world they find themselves, then that's generally a good thing. Normal genes, the normal function of genes is usually pretty good. The dysfunction is rare, but problematic. Those are the people who say, I'm not well, and go to the hospital, go to the physician. You can do that for epigenetic reasons too. The question you ask about what are the risks, this is a huge debate in medical genetics. So when I was chair of genetics at Case Western, in the department was a Center for Human Genetics, and we did both genetic diagnostic testing, and we trained genetic counselors. Now, if that condition was readily treated, like PKU, you change the diet, it's a devastating condition. If you change the diet, you can prevent the disease, and the baby, the child will have a normal life. So predicting for things like that is a really big deal because it improves life. If you knew that there was a genetic variant and you could change cancer risk by not smoking. Well, that could, and your risk was really high compared to baseline, you might change your lifestyle. We know if you knew you would be at risk for diabetes, metabolic disease, and exercise would be good, and the risk was really high based on that variant, then an individual might change their lifestyle. The way I phrased it is that knowledge is yours. It's your genetic information. This is my position about this, and others too. But it's your genetic information. If you want to know it, you should be able to know it. And not just a physician or anybody else, but it's your information. It's about you. Now, if you choose not to know, that's fine. But if you want to know, you should know. And then you can decide for yourself what you want to know and how you want to deal with it. Now, for some conditions like Huntington's disease, single gene, we can do the diagnosis. If your parent had Huntington's, you have a 50% chance of having two. There's nothing that can be done about that. It's a completely devastating disease and what we call non-actionable. There is no action that we can do given that information. But we can test. Some individuals who are at risk want to know because they say it changes the way they live, and others don't want to know because it would change the way they live. But it's up to that individual to decide. The other side is, what does society do with that information? Say I have a genetic variant that increases my risk for alcoholism. Just say. We don't know this, but say. And I choose, and there's health consequences for chronic alcoholism. But I choose to do anyway. And I incur health costs for the community, not just for me, but everybody else shares in the health cost. Do I have a responsibility to behave myself based on that information? Because not only impacts me, but affects everybody else too. These are real, and if you knew genetic information about you, by definition, you know information about a lot of your family too. And you can have some information about what your kids might be. So this isn't just your information, it's information that's related. So they're really, really interesting, hard, important questions. We're finally at the stage where but before we had the data, before we had the evidence, they were interesting but somewhat esoteric questions. Now they're really important questions. So what you ask is the center of medical genetics and genetic diagnosis and ethics. What's the right way to do this? To me, the fundamental principle is do it the right way, but make sure that the information is owned by that individual, and they can choose ultimately what they want to do with it.
You mentioned how in testicular cancer you can find heart cells, all sorts of.
Yeah.
Is that true for ovarian cancer as well?
It's also true for ovarian cancer. It's really weird. In the ovary, there's a strain of mice that gets ovarian cancer, and the eggs in the ovary act as though they've been fertilized. And so within the ovary, you can find 6-day embryos. If you hide all the ovary around, you say it's a perfectly normal 6-day embryo. And then they sort of lose their sense of organization and start to make muscle, skin, hair, bone, cartilage, brain. Sometimes these eggs are ovulated. And in virgin female mice, we can control that because the cage is only females. You can find pregnant females. So these eggs from the ovary have called diplodized. They've kept both copies. They haven't developed from gametes, so they're diploid. They're like genetically identical to their mom. They can implant. And we've seen 10-day embryos with the heart, eyes, beating, no dad.
Are they viable? Or?
No, they go to about 10 days because they need, even though it's genetically identical in DNA, it's epigenetically modified. So the DNA that you get from your mom is marked in ways that says, I'm from mom, and the identical DNA is marked, I'm from dad, and you need the balance of the two. If you're all mom or all dad, you don't live, even though the underlying DNA is exactly the same. So it's a special case of epigenetics. So they don't go to term. But the curious one is if the first mouse that was discovered like this way had gone to term, do you know what day it would have been born? Christmas. Okay? All right, thanks.